Hello, everyone. We're going to have another lecture on urinary physiology today. It's going to be our second lecture for urinary system. After this one, we're going to have another one for acid-base balance. Okay, so let me see. All right, so we're going to start urinary physiology. Let me stop this one and share my screen with you with the slides. Oops. Okay, so the first slide of this lecture is kind of a review for blood trace through the kidneys. And if you remember, the main function for urinary system is to filter blood. So we really need to understand how the blood get into the kidneys and come out, and the processes of filtering blood and also reabsorbing most of the materials back. So this is the trace, okay? Arteries from aorta, arteries branch out from aorta will form renal artery in the abdominal cavity and get into segmental artery, and then remember we took this one out, right? So segmental artery branch out to form interlobar arteries, which are in between the renal pyramids. And then interlobar arteries will form these arcuate artery, which is between the cortex and the medulla. Arcuate artery will divide and branch out to form a cortico-radial artery, also called interlobular artery. I think we should use the term from your book. It is called a cortico-radiate artery, okay? And then connected to the afferent arteriole, and then the first capillary bed, glomerulars, and then efferent arterioles, so blood coming out and going to another capillary bed. So it is either the peritubular capillaries or visa recta. Okay, if you remember, for juxtamedullary nephron, they have extended loop of Henle. So around the loop of Henle, the capillaries is called visa recta. Okay, and then the blood will be collected into cortico-radiate vein and merge to arcuate vein and merge to the interlobar vein and then renal vein. There's no segmental vein on this part. Yeah, please make a note on that. Okay, so the two types of nephrons, the functional and structural units in kidneys. This one is called a cortical nephron. Most of structures will be in the cortex, a little bit in medulla. But this type, juxtamedullary nephron, has very long extended loop of Henle into deep into medulla. We'll talk about the function of this today. And majority of the nephrons are the cortical nephrons. Only 20% of nephrons are juxtamedullary nephron. If we review the components of a typical nephron, we know there are 5 parts. If you can go with me, count these 5 parts. The first one is called glomerulars, and then Bowman's capsule, and then proximal convoluted tubule, and number 4 will be the loop of Henle. Number 5 is distal convoluted tubule. Yes, only these 5 parts. Okay, and all these nephrons, they are connected to the collecting duct, sending the filtrate to the collecting duct. So remember, all the fluid running in these tubular structures, they're not urine yet, okay? So there's still stuff being reabsorbed, being secreted, being excreted, and only when the fluid get to the end of the collecting duct, okay, getting to the minor calyx, then it's urine. Okay, so the details, cellular details for each part of the nephron. Okay, so of course we know glomerulus is lined with simple squamous epithelial cells, and also we talk about the visceral layer of Bowman's capsule is made by podocytes. If you remember, the podocytes, they have extended pseudo arms and kind of like intercalated fingers, all right, so to form the filtration slits, okay, and then the simple squamous epithelial cells from glomerulus, basement membrane, plus the filtration slits, all three parts together to form the filtration membrane. Okay, that's where the blood being filtered through from blood capillaries into the Bowman's capsule. All right, so if we look at the proximal convoluted tubule, the lining facing the lumen here, we mentioned this last time. This is a review, okay? It's by simple cuboidal cells. The nuclei are kind of closer to the apical surface. There are many long microvilli here to increase the surface area. On the basal side of the membrane, there are many mitochondria. Okay, mitochondria is the powerhouse. The function is to make ATP. So if you see a lot of mitochondria here, that means there are a lot of active activities, active activities using ATP. That means using ATP on this side of the cell membrane. If you look at the loop of Henle right here, okay, loop of Henle, and you don't need to know too much details about the lining of ascending and the lining of ascending and lining of descending the loop of Henle. You need to know in general the loop of Henle is lined with simple squamous epithelial cells, so it's pretty thin, okay? And then we get to the distal convoluted tubule here. The lining here, similar cuboidal, simple cuboidal cells, but comparing to the PCT, much less microvilli, okay? Nucleus are more centered and less mitochondria on the basal layer, basal side. Okay, if we look at the collecting duct here, all right, so almost no microvilli here and very, very few mitochondria. So here it indicates that there are not very much of active activities in the collecting duct, mostly are passive. Okay, so now if we look at this area, so if you think about this area, what is this area called? Let me give you choices. Okay, this circle right here representing A, glomerulus, B, Bowman's capsule, C, PCT, D, DCT. Which one is it? It's PCT, right? PCT. So proximal convoluted tubule, right connected to the Bowman's capsule. So here, all right, enlarge this PCT area. We can see on this side is the peritubular capillary, okay? So this is the blood capillary. And then on this side is the lining cells, simple cuboidal epithelial cells, all right? And here is the lumen. Here's the lumen here, lumen in the PCT, okay? So blood has been filtered in the glomerulars into the Bowman's capsule, and then the filtrate start to travel in the PCT. Okay, while the filtrates traveling down the PCT, many of the materials get reabsorbed back from the lumen to the blood capillaries. Okay, so this part, okay, is the materials traveling from lumen back to the blood is called reabsorption. All right, PCT is the major site for most of the materials reabsorbed back to the blood. Okay, so if we talk about glucose and amino acids, all of the glucose molecules and amino acid molecules are reabsorbed at the PCT. If there's any glucose, let's just use glucose as an example. If any glucose past the PCT and not being reabsorbed, then you're going to see sugar in urine. Okay. Another word, PCT is the only area for glucose reabsorption. Okay, so for a normal person, of course, all the glucose molecules will be reabsorbed back into the blood at a PCT. So there's no glucose molecules in urine. Okay, amino acids are reabsorbed very similar way as glucose. So we just use glucose as an example for reabsorption process. You do need to understand this process and be able to explain this process in your own words. So let me give you this process. All right. So on this diagram here, there's some arrows indicating different types of transport. If it's a solid red, means primary active transport. So do you remember what is a primary active transport? That's the pumps, okay? Sodium, potassium pump, hydrogen pump, calcium pump. If it's a pump, then it's active transport. It requires ATP, okay? If it's a dashed red arrow, it means a secondary active transport. So if you forgot what is secondary active transport, you may want to go back, read your textbook a little bit more, from the early 430 lectures. Okay, so basically here you see the secondary active transport. It is still active transport, so it is still requires ATP put into this process, but it's a secondary active transport because it has two parts. The first part requires ATP, the second part does not. Okay, so the total process have two steps. The first part requires ATP, the second part does not. Okay, together we still say, yeah, there are ATP required for this process. If it's a blue arrow, it is a passive. Okay, passive. That means no extra ATP required for the process. The simple diffusion or facilitated diffusion, they're all types of transport, passive transport. Okay, all right, so with this in mind, we can explain how the glucose is being reabsorbed into blood. Again, this side is the lumen, the filtrate is traveling this way. Okay, these are the lining cells for PCT, and on the side there are blood capillaries, peritubular capillaries. Okay, so this side we call is the apical surface facing lumen. This side is the basal membrane facing the blood capillaries. So if you look at the basal membrane, basal layer, basal membrane over here, okay, there are pumps, solid red arrows. So this one is a sodium potassium pump. Okay, pumping sodium out, pumping potassium in against the concentration gradient, right? So it's pumping sodium from low to high, pumping potassium from low to high, okay? And then you see on this side of the membrane also have this passive channels for potassium. These are leaky channels. If you see here, passive transport. These are leaky channels. So potassium will continue to leak out if there are more potassium inside of the cell than outside of the cell. So this mechanism right here, you continue to pump sodium out, okay? So pump sodium from low to high. Continue to pump potassium from low to high. But once the potassium gets in, because the higher potassium concentration here than outside, potassium will be leaked out by leaky channels. All right, there's no sodium leaky channels on this side. Okay, so sodium will continue to be pumped out. So think about inside of this cell, the sodium concentration is low. This is what you want. You want to create a very low sodium concentration area inside of this cuboidal cell. This is low sodium concentration here comparing to in the filtrate right here in the lumen. Lumen, when you first filter out blood, there are a lot of sodium ions in the filtrate. So sodium concentration is very high in the filtrate. And here, because you keep pumping sodium out inside of this cuboidal cell, the sodium level is very low. Now you have a huge sodium concentration gradient, which will have a driven force for sodium to move from the lumen into these cuboidal cells. Okay, so that's why you see this one. Sodium is moving in. This is a passive right here. It's a diffusion. It's a passive. Okay, it's a facilitated diffusion because it's ion. It's ions. It needs to have carriers, but still, it's moving from high to lower concentrated areas. So there's no ATP, extra ATP required for this process. Okay, but in this moving transport, in this type of transport, when sodium is moving into the cuboidal cell, it at the same time it drags a glucose with it. So in other words, this carrier not only carries sodium in, but also can carry a glucose molecule in. All right. So remember when the blood being filtered out, all the glucose molecules will get into the filtrate. Now it is the time for glucose being reabsorbed back toward to the blood capillaries. How can glucose get in? Is based on the sodium movement. Sodium movement drags glucose in. Without this sodium movement, glucose cannot be getting back to the cuboidal cells. All right. Once the glucose get into the cuboidal cells, and then it can move toward to the blood capillaries. This part of transport of glucose is all facilitated diffusion from high to low concentrated area. So the key for glucose reabsorption is this part. Okay, first come in from the lumen, from the filtrate into the cuboidal cells. But this part of glucose transport, is solely depend on the sodium movement. How can we make sure sodium will move from filtrate to the cuboidal cells? It is depend on this part because you have a sodium potassium pump. Continue to pump sodium out to make a very low sodium concentrated area inside of cuboidal cell. This one will allow sodium to move from the lumen into the cuboidal cell. All right, so let me summarize this process. So the whole purpose for this mechanism is to move glucose and amino acids back into blood from filtrate. To do that, you need to have separate steps. Number one is on the basal membrane, this side, okay, there are sodium potassium pump, which works constantly to pumping sodium out, pumping potassium in. This one definitely is active transport, requires ATP. All right, so the sodium concentration will get lower and lower inside of the cuboidal cell. At the same time, the potassium concentration, if you just have the pump, okay, you're going to build up potassium concentration inside of the cell. But you want to balance that. You don't want to build up too much potassium in here. So this balance can be done by using these leaky channels. There are potassium leaky channels on the basal membrane. So potassium can continue to leak out as long as there's a potassium concentration gradient across this basal membrane. Okay, so this pumping activity results in a very low sodium concentration in the cuboidal cell, and this will give a huge concentration gradient between the filtrate sodium and sodium ions inside of a cuboidal cell. So this sodium concentration gradient is the driving force to allow sodium to move from filtrate back into the cuboidal cell. This movement, the force is really strong because the concentration gradient is really big. Okay, so when sodium is moving toward to the cell, it drags a glucose with it. That is called a co-transport. So one transport system transporting two molecules, two different materials in. So this is a co-transport. This part of transport does not require extra ATP because it moves sodium from high to low and drags the glucose in. Okay, so this is called a co-transport. This is the second part of the secondary active transport. So if you look at active secondary active transport, it has two parts. The first part is the primary active transport, the pumps. The second part is facilitated diffusion. Okay, this part one plus part two together is called a secondary active transport. Okay, so we say all the glucose molecules and amino acids can be actively transported back or reabsorbed back into blood by secondary active transport system. Okay, so the key for glucose reabsorption is to pass this apical membrane, get into the cuboidal cell, and then from the cuboidal cell out to the extracellular space and then into the blood capillaries. These parts are all done by facilitated diffusion. It's easy, all right? So this part, I really want you to understand, you do need to understand how glucose molecules being reabsorbed from filtrate back to the blood capillaries. Okay, so you really should review this process and write down this process in your own words. Once you understand the glucose transport, then you should be able to understand amino acids transport because they're using very similar mechanisms. So this part of transport is done in which part of the nephron? PCT only. Yes. Okay. Besides of glucose and amino acids, majority of water and ions are reabsorbed in this area as well. That's PCT. Okay, PCT. Now we're down to this loop of Henle, the function of the loop of Henle. Okay, this part is a kind of confusing concept. So I really need to have your attention for this part, the function of loop of Henle. Okay, so loop of Henle, of course, there's an ascending part, there's a descending part. So here is the descending part. Here's the ascending part. So this is all loop of Henle structure. Okay, so remember here is descending closer to the PCT, ascending away from PCT. Okay, that's a loop of Henle. So on this diagram, we use this U-shaped loop to representing the loop of Henle. So this is the descending, this is the ascending loop of Henle. Okay, so first let me explain the channels and the fluid movement around loop of Henle, and then I'll tell you why it is important here. So we're starting with the ascending part of the loop of Henle. Okay, on this part, you need to remember, probably you should write it down. Ascending part of loop of Henle is impermeable to water. Impermeable to water. No water can move out from the ascending loop to the surroundings or move into the tubular structure. Impermeable to water, right here. Impermeable to water. But ascending part is permeable to ions. To ions. So you see the potassium, sodium, chloride ions, they're all can move in and out, right? But here, these are solid red arrows. That means these are active transport. So these ions can be actively transported out from the loop of Henle, ascending part. Remember, ascending part of loop of Henle? Okay, this part of tubular structure is lined by simple squamous epithelial cells. Okay, so there are active transport systems for ions here. Now we look at the descending part of loop of Henle. Okay, write it down, please. It is only permeable to water. Only permeable to water, not permeable to ions. All right, so now you see the difference. The ascending part only permeable to ions. The descending part is only permeable to water. Okay, keep that in mind. All right, now we can talk about the mechanism. Step by step. Let's say we stop the kidney functions, we restart from the very beginning, restart this filtration process. Okay, so this is the loop of Henle, descending, ascending. Okay, so in this yellow tubular structure is the filtrate coming in. Okay, so this 300 representing the concentration, the total osmotic concentration. So just to remember, it's a 300 units. Okay, this solution has a certain concentration, is a 300 osmoles, or you just remember it's a 300 units. Okay, units for concentration. All right, so we start from this ascending part. Remember, it's only permeable to ions, impermeable to water. Okay, so these ions can be moved out from the tubular structure into the surrounding area by pumps. So you're pumping these ions out. So once you're pumping the ions out, think about the concentration here in the ascending part is getting more concentrated or less concentrated. less concentrated because you're pumping the ions out, right? So the surrounding area, the concentration is increasing. Okay, at the beginning, they are balanced, equalized, right here, equilibrium. All right, so now you start pumping ions out, then the surrounding area will have more concentrated solution than inside. So if you start from 300 units and it's going to gradually getting smaller, maybe 200, 150, things like that. So at the beginning here, when the filtrate gets into the beginning part of ascending, a lot of ions being pumped out because you have a lot. And then when the filtrate continue to move up, and the less ions will be pumped out, but still going to pump out, right? You have less and less being pumped out because the concentration is decreasing. You got that, everyone? Yes? Okay. So that's at the very beginning. And then the next, this is a continuous process. We just break it down into steps, try to explain what is going on here. All right. So you see here is 200, here's 300. What is going on here? Because the ions being pumped out. Okay. And around this tubular structure, it was 300. Now it's more because you add more ions in. Okay, so the concentration decreases, all right? And then because this area around the tubular structure, now the concentration increased from 300 to 400, 300 to 400. Then this filtrate coming in is only 300, okay? So now, which side has more water? Of course, this side will have more water than this side, right? This is less concentrated. That means more water in the descending part than the outside. See this concentration difference? Okay, because you have more water in the descending loop. Remember, descending loop is permeable to water. Only permeable to water. So even though there are more ions here, ions cannot move back. It's impermeable to ions. Only water can move. So water will go from a higher water concentrated area to the lower water concentrated area. That is called osmosis, if you remember that. Okay, so this green arrow representing osmosis is the pure water movement. Water move from a higher water concentrated area to the lower water concentrated area. So if water start moving out from descending loop, think about this 300 concentration, total concentration. Is that going to become, this filtrate, is that going to become more concentrated or less concentrated? Start from 300. You're moving out water. Then this filtrate will become gradually more and more concentrated. So entering this loop of Henle is 300. Once it gets to the end of this descending part, is going to be much more concentrated than 300. Okay, so this kind of movement is a continuous. Okay, so start from 300 and then you're going to pump out ions and then moving out water. Okay, so this process will continue until it gets to an equilibrium. Okay, once it gets to the equilibrium, you'll actually create a system. It's kind of a stable. Once this concentration numbers showing like this, it is a stable condition. That means this this movement is reach the equilibrium. Okay, the ion moving out, water moving out, and the filtrate movement get to the point it's stabilized. All right, so at this point, okay, you can see this concentration changes. All right, filtrate coming into the loop of Henle, it starts with the 300 milli osmol. Okay, and then this 300 unit concentrate is 300 unit solution will gradually increase in concentration because osmosis moving water out. Once this filtrate gets to the end of a descending loop, it's 1400 now. Okay, comparing to 300. It's really, really, really concentrated. Okay, so a lot of water moving out. Okay, and then when this concentrated filtrate start going into the ascending loop, all right, the ions will be pumped out from loop. Okay, so now through this process, you can see a lot of water will move into the surrounding and eventually getting to the vasa recta, the blood vessels. The ions will move in here and get into the blood vessels. So water and ions are reabsorbed back. Okay, and also if you look, the starting of the filtrate is a 300, and when this filtrate leaves the loop of Henle, it's only 100. It's much diluted. Okay, because most of the ions are reabsorbed back, and also water. So start with a lot of volume, but here is much less volume and much less concentrated. Okay, so that's one key point. The second key point is more importantly, okay, because of this process, you create a concentration gradient in the medulla, all right, in the medulla area. Okay, so if you look at this color changing from light to dark, representing the concentration changes at the deep part of the medulla is 1400, and at a medulla close to the cortex is only 300. So you create this concentration gradient around this loop of Henle. This part has a very significant functional aspect for reabsorption. I'll explain this in a minute. Okay, so first let's look at this process. Make sure you understand why we end up with this concentration gradient in the loop of Henle and also in the surrounding medulla. Okay, think about it over and review this process and write down all your questions so we can discuss this more in office hour. Okay, so now this is the end result. Okay, when this filtration process gets stabilized, then the filtrate will go through the loop of Henle and then create this concentration gradient in medulla. So you see this filtrate has the changes in concentration, and now you know why. Okay, and then you have this concentration gradient established in the medulla. Okay, this mechanism is called a countercurrent multiplier. Countercurrent multiplier. So if you need a little bit more assistance in understanding this concept, you may want to go to YouTube and find some short videos about, talking about the countercurrent multiplier. And also your book will be a good resource. Reread this part to understand it, okay? All right, so now we have this in deep of medulla, you have this established the concentration gradient. So this is, we only talking about the juxtamedullary nephrons because only this type of nephrons have extended a long loop of Henle into medulla. Okay. All right, so now we can talk about the function of loop of Henle. Okay, so loop of Henle allows production of concentrated urine. You said, wait a minute, the urine leaves the loop of Henle actually is more diluted, but remember the volume wise is much, much, much less, right? Most of water will be reabsorbed. And so that's why I minimize water loss right here. Okay, and in terms of function here, okay, ion permeability, ascending, is that ascending permeable to ions? Yes. Descending, no. How about water? Ascending, no. Descending, yes. Okay, so osmolarity comparing to each other, is ascending more concentrated or descending more concentrated? Yes? Okay. So the loop of Henle, the longer the loop of Henle, can we say the more water and ions being reabsorbed? Then will be less urine. The longer the loop of Henle, the more water will be reabsorbed. Then it will be less urine. So if you think about the desert animals like kangaroos, right? If you look at their kidney structures, they have more juxtamedullary nephrons. They have much more longer extended loop of Henle. So that means they can reabsorb water more to minimize the medulla here will be most concentrated. This part will be least concentrated. Okay, so why is that important? Okay, now think about this collecting duct right here, collecting duct. So all the filtrate will get into the collecting duct. Okay, when this collecting duct are traveling from here, the beginning of the medulla, all the way to the deep of the medulla and then connected to the minor calyx. Here, this filtrate is traveling in this duct, but the surrounding of this collecting duct, the concentration changes, okay, from low to high. So look at this. All right, so the where am I going? Right here. So this is the loop of Henle, okay? So this filtrate going to the collecting duct is 100. It's around 100 concentration units. So once you get to the collecting duct, think about around this collecting duct, the concentration is from 1200 to 300. It gets in is only 100. Okay, it's much diluted than the surrounding. So because of that, water will continue to move out. Water will continue to move out, okay, as osmosis. Okay, and so the urine here start from 100 units, will continue to be concentrated at did I say filtrate or urine? The filtrate will starts with 100, will continue to be concentrated, and then once it gets to the minor calyx right here, is 1200 units. This is urine. So by moving from this collecting duct, from the beginning of the collecting duct to the end of the collecting duct, more water can be reabsorbed back, right? So at the end, when you talk about the urine, it's a very small amount in terms of volume, comparing how much blood being filtered, the volume of filtrates. Okay, the urine is a very small amount. Okay, so this is the importance of the loop of Henle for juxtamedullary nephron, because of this nephron function is going to create a concentration gradient here from 1200 all the way to the 300 in medulla. This will allow water continue to move out from collecting ducts. Okay, your book also talk about the urea is a small protein which helps to create or establish this concentration gradient. You can just skip that part. We just talk about the ions and water movement. We will skip the urea's function in this area. Okay, so the water has ability to move out in the collecting duct. This is very important. If you look at this side of the diagram comparing to this side, if there's no water moving reabsorption in the collecting duct, see, you start with 100 unit solution here, filtrate, same thing, but this one will continue to get out from collecting duct. But this one, you're going to reabsorb a lot of water back, all right, and have a concentrated urine. So comparing this to this one, you're going to lose a lot more volume in urine. This is not a good condition, because if you lose too much volume, then your blood pressure will be low, right? You don't want that. You just want to get rid of unwanted stuff, okay? So you want to reabsorb most of the water, minerals, and all of the amino acids and glucose molecules back, all right? So you have a concentrated urine right here. So collecting ducts and the distal convoluted tubule, these are the two areas for hormone regulation. If you remember the antidiuretic hormone, the function is to increase water reabsorption by increasing the water channels. All right, so the functional site, the target site, is collecting duct and DCT. Okay, and also aldosterone. If you remember, aldosterone made by adrenal gland. Okay, aldosterone's function is to increase sodium reabsorption. When sodium goes, water always follow. So by increase sodium reabsorption, it will reabsorb more water as well. So antidiuretic hormone and aldosterone, these two hormones function on collecting duct and DCT. Okay, the end result is the same. It is for water reabsorption, but the mechanisms are different. For antidiuretic hormone is directly increasing the water channels for water reabsorption. For aldosterone, okay, it increases the sodium reabsorption and and then water follows that. Okay, this is the same slide. Okay. All right. So now we talked about the structures here. The filtration part, filtration membrane from this glomerulus and Bowman's capsule. And then we talk about PCT here. We say most of this materials will be reabsorbed here. Most of the water, most of the minerals, and all of the amino acids and glucose molecules. So this is why you have a long microvilli to increase the surface area, so you have more carriers, okay, to reabsorb glucose and amino acids back. Okay, and then if you look at the loop of Henle here, simple squamous epithelial cells, and then remember there's a difference for descending and ascending parts. All right, so these descending, ascending parts, because the permeabilities are different, okay, this will help to create a concentration gradient in medulla. This concentration gradient will help the filtrate in collecting duct to reabsorb, to have more water reabsorbed back. So the final urine is very concentrated, but small amount of material, okay, liquid. So the distal convoluted tubule right here, okay, distal convoluted tubule right here. This one is mostly for water reabsorption, kind of like the collecting duct, okay? So no active transport anymore. That's why you don't see a lot of mitochondria over there, and not a lot of microvilli either. Okay, so the filtration is very important. We want to keep a stable filtration rate. Okay, it's called a glomerular filtration rate. That means the filtration, you want to have a very stable filtration process inside of renal corpuscle. Okay, so to regulate the filtration rate, okay, there are two big arms of regulation. One is local regulation. We call it intrinsic regulation of glomerular filtration rate. This is auto regulation. Another one is called extrinsic regulation by changing systemic blood pressure to regulate the glomerular filtration rate. So we can explain this, all right? So first, this one, if you see right here, this is Bowman's corpuscle, has the Bowman's, renal corpuscle, has a Bowman's capsule here, okay? And two blood arterioles. The one going in is afferent, one's coming out is efferent, right? Arterioles. And then you can see the afferent arteriole is bigger in diameter than the efferent. That means more blood coming in, a delayed blood going out. This will create a pressure in these glomerulars to allow the filtration takes place. So filtration membrane is right here. Do you remember the filtration membrane right here? Okay, alrighty. So if we look at the structure here, this side is connected to the PCT. Okay, PCT, long microvilli, right here. Yeah, simple cuboidal lining. All right, This side, okay, is a cross section of DCT, distal tubule. Some book will say this part is a kind of like end of ascending loop of Henle. It doesn't matter. There's no clear cut. We just say this is a cross section of the beginning of the distal convoluted tubule. Okay, so this part of the tubular structure, they're always very close to the arterioles. If you look at this cross section, okay, see these are typical cuboidal cells with hardly any microvilli, but there's a part of this tubular structure, these cells facing the arterioles, they are specialized. They're more like a simple columnar cells. Okay, these cells are chemoreceptors or chemosensors. They can sense the sodium concentration changes in the fluid inside of this DCT. Okay, so they're chemosensors. All right, if the sodium concentration changes in this tubular structure, then they start to releasing chemicals. Okay, so we'll talk about that. That is part of intrinsic mechanism. So along this afferent arteriole, there are cells around it. It's called juxtaglomerular cells. In your book, it's also called granular cells. Same thing, okay? These granular cells, they can release renin. Do you remember renin? Renin-angiotensin mechanism to regulate the blood pressure. Okay, so the renin, the hormone renin, is released by these granular cells or juxtamarular cells. Okay, so granular cells, macular denser, and also there's another type of cells is called mesangial cells. They're kind of like a fat cells filled up in this space. Some people say these cells can transfer signal from macular denser to the granular cells. Okay, the real function of these cells we still don't know. All right, so in this filtration structure, filtration apparatus, okay, we have these granular cells, macular denser cells, and mesangial cells. These cells in this local area form a functional structure. It's called juxtaglomerular apparatus. Juxtaglomerular apparatus. This is the local regulation to regulate the glomerular filtration rate. Okay, so let's say, if less, if how should I say? Okay, if blood pressure is lower, then the flow of blood going to the afferent arteriole will be lower. All right, maybe let me wait. Okay, maybe I have another slide. Yeah, this one. Let's use this one to explain the local regulation. This is much better. Okay, so let's look at this side. This blue and orange, this part is intrinsic mechanism, and the green and the yellow is extrinsic mechanism. Okay, both of these arms are going side by side, but the intrinsic mechanism is more like a local, second by second regulation, and extrinsic mechanism is a stronger regulation by changing the systemic blood pressure. Okay, so the scenario is blood pressure drops, and then that's going to lower the glomerular filtration rate. So to adjust that glomerular filtration rate back to normal. These are the mechanisms. So we were talking about this role here. So let's just pick up this one. Okay, the systemic blood pressure is lower. Okay, and then the filtrate flow is going to be slower. Yes, and then the juxtaglomerular cells and also macular denser cells will sense the changes. Okay, so macular denser cells, they are chemical sensors. Remember? Let me see. Let me find the. Okay, this is in your book 988. Okay, so then if you have a lower flow to the nephrons, then you have less sodium concentration going through the tubular structure. Then the macular denser cells will promote to release of vasoactive materials to change the diameter of the afferent arterioles. Okay, let me repeat this one more time. Okay, low blood pressure in the blood vessels. Okay, less filtrate flow through the distal tubules. This less filtrate will be sensed by macular denser cells. Less filtrate, less concentration, less sodium, right? And then the these changes will be sensed by chemo sensors, and then lead to the release of vasoactive chemicals, and then it's going to dilate the afferent arterioles to allow more blood flow into the area. Okay, let me see this one. Okay, so right here, start with less flow into this nephron structure, and then the filtrate in the distal convoluted tubule. Okay, less flow, less sodium can be sensed by these macular denser cells, and then these changes will lead to release of vasoactive materials. These vasoactive chemicals will allow this afferent arteriole to dilate, to increase the diameter, to allow more blood coming in. All right, so you got that? All right, okay, so that's this side. Okay, and also intrinsic regulation has something called myogenic responses. This is autoregulation as well. Myogenic response is a part of the property for smooth muscle cells. Basically, it says the more stretch of the smooth muscle cells, the stronger the contraction. That's called myogenic response. And if you look at this area, this along these arterioles, especially along these afferent arterioles, there are smooth muscle cells around. Okay, so if more blood coming in, you're going to have these smooth muscle cells stretched, then it will cause a stronger contraction to decrease the diameter of this arteriole to minimize the blood flow in. Okay, but if you have less blood flow in, you have less stretching and less contraction, so relatively bigger diameter to allow more blood in. Okay, that's called a myogenic response. So myogenic response and also the macular denser response, these are the auto-regulation part. It's intrinsic mechanism. All right. For the extrinsic mechanism, is the goal is to by changing the systemic blood pressure to adjust the glomerular filtration rate. So if you remember the angiotensin mechanism, right here, angiotensin mechanism, one main hormone involved in this mechanism is renin, right? So now we know renin is released by granular cells right here. Okay, so by releasing granular cells, by releasing renin by granular cells, it can change systemic blood pressure and then change the glomerular filtration rate. So this part we already talked about in the early Unit 3 lectures, the blood pressure regulation. So the green part will be the hormonal regulation, the yellow part will be the neural regulation. But overall, the whole purpose is to change the systemic blood pressure to regulate the glomerular filtration rate. Okay, so we're done with the physiology part. And then last time I did tell you, you are responsible for the structure of the urinary bladder. Okay, so the lining, the structure listed on your lab handout, you are responsible for that. And I just want to mention there are two sphincters around this urinary bladder. This is external sphincter, internal sphincter. External sphincter is made by skeletal muscles. So this part is voluntary controlled, so we can control when we want to go to the bathroom because this part is somatic controlled. But you think, the babies, they have to go through the party train because this part, we know is somatic, but we still need to train the babies to get the connections from the cerebral cortex. Once you have these connections established, then you can control when to open or close this external sphincter. This is the strong sphincter comparing to this internal sphincter. Internal sphincter is made by smooth muscle cells. Okay, it's relatively weaker sphincter. All right, so I have a few of histology slides, and you can also find these in my lab and a mastering site. Okay, so this one definitely is a kidney structure. How do I know that? You see these parallel structures. That's the renal pyramid. All these collecting ducts. And see here, you don't see that. That is the cortex. All right, cortex will have the renal corpuscles and also a lot of convoluted tubule. Okay, and then this one is showing the Bowman's capsule with glomerulars. So this is Bowman's capsule. You can see it is connected to the PCT. So this is the part of PCT. Okay, you see a lot of microvilli over here, simple cuboidal cells. Okay, if you compare this type of lining to this, see much bigger lumen, flat cuboidal cells, no microvilli. This is more likely is the cross section of collecting duct. All right, and then you see something in between, like this one or this one. Okay, so it's not as many microvilli like this one, but is less lumen, maybe a more square looking cells comparing to this one. And so this probably is the cross section of DCT. Okay, so of course these tubular structures, you say exactly where is the clear cut. It's hard, all right? It's gradually changing. So if I ask you to identify um, the tubular structure, I will use very typical structures instead of finding something like vague or in between. Okay. So again, this one is a collecting duct. This one is a cross section of a PCT. PCT. Okay, PCT here. All right, this is a longitudinal cut. Longitudinal cut of a PCT. All right, see the microvilli over here. All right, this is the, do you know what type of lining is this? Everyone? Lining of this? Is this epithelial cells first? Yes? Because you have a free lumen, right? Okay, so this is a transitional epithelium. Transitional epithelium. And where can you find this type of lining? Urinary bladder. And part of the urethra. Okay, so, all right. This one is, I did label this, okay? So kind of give you an idea of the juxtaglomerular apparatus, okay? So this is the renal corpuscle, all right? And you have the glomerulars, you have the Bowman's capsule here, connected to PCT, PCT. So on this side is always connected to the tubular structure, so we call it a renal pool. Okay, so if you have a PCT on this side, you know right away the arterioles should be on this side. Arterioles should be on this side. And then if arterioles on this side, remember this DCT close to there and side of the cells facing the arterioles are specialized macular denser cells. Okay, so let me go back and maybe I'll use the slide to explain this right here. Okay, so on this side is always connected to the PCT. We call it renal pool. Okay, so opposite side here, you always have the arterioles, and we can call it arterial pool or vessel pool here. Okay, if you have arterioles on this side, and then this macular denser cells always facing the arterial side. You got that? Okay. Okay, so now these are macular denser cells, cross section of DCT or the last part of ascending loop, okay, macular denser. And then in between here, right here, those are mesangial cells, okay? And here's the PCT. Same slide. Okay, this is the slide. This is a model we have in the lab, and I checked on the My Lab. They have similar, very similar one online as well. So I just want to use this to explain this model a little bit. Okay, so here is the cortex. Number one is cortex, all right, and number three here is medulla. So you see the parallel tubular structures. Oops, what did I do? Okay, and then if it's a deep, if this juxtamedullary nephrons extended tubular structures into the medulla, then along this loop of Henle, you have the capillaries. That is called, number 6 is called, no, not number 6. I did not label this. So this part of capillaries is called, yeah, that's what I want to label. But this one, sorry, oops. I put the arrow in the wrong place, not number 6. Number 6 is labeling the collecting duct, right? So this part of the capillary is called vasa recta, yeah. And this part of capillary right here in the cortex between on the side of tubular structure is called the peritubular capillaries, okay? And then this is the artery separating or right at the edge between cortex and the medulla. It is called arcuate artery. And then this artery going extended into the cortex is called cortical radiate artery. Okay, so from cortical radiate artery branch out into the renal corpuscle. This is afferent arterial. This one coming out is efferent arterial. Afferent, efferent, afferent, efferent. Can you see the size difference? Yes. Okay, all right. And then you can trace, same thing here, afferent, efferent, okay? And then you can trace the tubular structure. So if it's on the opposite side of the arterioles, this side, this orange color tubular structure has to be proximal convoluted tubule. And this, so you can see proximal convoluted tubule extended to become loop of Henle and coming up. Then this purple one, purple tubular structure close to the arterioles, it has to be distal convoluted tubule. You got that? Okay. Number 7 is close to the end of the collecting duct. The collecting duct at the end will be connected to the minor calyx. You got it? All right. So this is the end of this lecture. Okay. And we're going to have the third lecture for this week, which is going to be the acid blood pH regulation. Blood pH regulation. So that will be the next lecture. All right, I'll see you next time, everyone.